Everyone, my name is Lize Engelbrecht. I'm from Stellenbosch University in South Africa, and I run the confocal microscope at the university. Today, we will be talking about the resolution of light and its limitations. Let's quickly look at an overview. We'll talk about what resolution is, the differences between optical resolution and spatial resolution. We'll also talk about wavelength of light and the numerical aperture of the system and how it affects the resolution, and how airy disk, the diffraction pattern, and point spread function is related to resolution. We'll then also, of course, talk about the limitations of optical resolution and move on to spatial resolution and how pixel size and the Nyquist theorem affects the spatial resolution. And finally, we will talk a little bit about practical considerations. If we talk about what resolution is, we usually don't ask ourselves, what resolution should I use for my project? We ask questions like, what are the smallest features that we can visualize? How can we see the finer structures in my sample? Or how can I achieve more crisp images? Can we distinguish the different features in my sample? Or what are the features that I need to visualize and their size? All of these questions are related to the fact that resolution is important. The formal definition of resolution of an optical microscope is basically the shortest distance between 2 points in a specimen that can still be distinguished as two separate entities by the camera or the observer. It's important to also consider always whether you really need the highest resolution for the question that you ask in your research experiment. Let's look at optical and spatial resolution. Optical resolution refers to the inherent ability of the microscope or your imaging system to distinguish the fine details of your object. It's dependent on the physical properties of the microscope, like your objectives, the numerical aperture, all the optics of the system. When we talk about spatial resolution, it refers to the detail or the level of detail that is captured in your digital image, which is always determined by the pixels that you use to represent that image. So it's a property of the digital image produced by the microscope. Let's talk about optical resolution first. So optical resolution is mainly limited by the diffraction of light through your imaging device or your microscope. There are two aspects directly influencing the optical resolution, and that would be the wavelength of light traveling through the system, as well as the numerical aperture of the system. The numerical aperture of the objectives and the condenser is important here. You will see here in the equation of resolution that wavelength and numerical aperture are the two things that plays a major role in the resolution that can be achieved. When we look at the wavelength of light, it's important to remember that wavelength light is part of the electromagnetic spectrum, which means that all of the different colors of light have a specific wavelength. You get light that has shorter wavelengths, which means they have higher frequency and higher energy. And then you get light that has longer wavelengths, lower frequency, and therefore lower energy. This is related to the color. So your violet light usually has a wavelength of around 400 nanometers, which is the shortest wavelength. And then your green would have something like 550 nanometers and red 665 nanometers. This means that all of these wavelengths of light have different energies. They refract differently through the system, and therefore they also can only achieve different resolutions. Your violet usually have a little bit higher resolution than the red, being a shorter wavelength. Another aspect, the numerical aperture. So on an objective, you usually get the number of the magnification, which we're all familiar with, 100 times, 60 times, 40 times. But there's another number which is called the numerical aperture of the microscope. This number can be found by this equation where two aspects is important: the maximum angle of acceptance of light from the sample's diffraction pattern, as well as the refractive index of the media that is between your objective and your specimen. If you have an objective with a low numerical aperture, the angle of that diffraction pattern that can enter into the objective is much smaller than your higher numerical aperture objectives. A high numerical aperture can capture a much wider angle than your lower numerical aperture objectives. Another aspect is the refractive index. if you look at this sample where light travels through the slide, your sample and cover slip, there's only air between the cover slip and the objective, which means that the light refracts wider than the objective can capture. This means that this is a lower numerical aperture objective. If you have a higher numerical aperture objective, it usually has an immersion of, for example, oil. And you can see that the light that passes through the whole system does not refract away from the objective and is actually taken into the objective. And that light contributes to the resolution. If you need more information on the numerical aperture, there's some course video that you can have a look at. And there will be other presentations in this series that will also talk about objectives a little bit more. The airy disk is basically a pattern that forms if you look at the fluorescence light of a single particle that travels through the microscope. If I show you here, the light of a single molecule, for example, will travel through your objective and form this pattern in the back of the system, where the center is a very bright center with some minor rings around it. If we represent this pattern in its intensity, you'll see that the minor rings are much lower intensity than the center peak. And if we represent this as a three-dimensional intensity peak, this is what it would look like. The reason for this is because there's diffraction throughout the objective. The diffraction pattern is formed at the back aperture of the objective when light travels into the objective. You will see that there's a major peak right in the center. This is called the airy disk. And this is also the width of this airy disk is determining what we call the point spread function. The point spread function is basically a three-dimensional representation of the diffraction pattern of a single fluorescent particle. In other words, if there was a molecule, a fluorescence molecule, and its light has passed into the objective, this is the pattern that you would get without its out of focus light. But if you include the out of focus light, this is what the pattern would look like. For more information on the diffraction pattern and the point spread function and how this affects the numerical aperture, there's another course video as well as an interactive tool that you can go and have a look at. The optical resolution in the system always have a limitation. And what determines this is basically, as we said, the diffraction pattern. So what does it look like? If you have a single emitter, single fluorochrome or a single bead, the smallest that you can visualize that bead is usually about 250 nanometer, give or take, depending on the wavelength of that light. This is much bigger usually than the emitter. If you have a structure that consists of a number of these emitters, which is all of them together smaller than 250 nanometers, they would basically also give a very similar pattern than your single emitter because the resolution is limited. In an image that we look at, where we have multiple emitters or fluorochromes emitting light, you sometimes can resolve all of them because they are spatially separated. But at some point, if they are starting to get too close together, you cannot resolve those separate fluorophores anymore. So in a biological sample like this one, which is a cell, you can see here the nucleus is stained in blue. You have the red mitochondria, and then in green, we have actin filaments. We cannot really see the details inside the mitochondria due to the fact that it's smaller than the resolution limit of the microscope. Also, actin filaments, it might look like it could be a single strand, but we do not know that because if the resolution is limited, some of these strands might actually consist of a lot more strands. We also do get super resolution microscopes that improves that resolution to some degree, where you can see more clearly the single mitochondria here, but it's still fairly limited in optical microscopy. Other aspects are also influencing the optical resolution that you can achieve. If you do not have good enough contrast in your specimen, so low specimen contrast, or if there's not proper illumination of the sample, that will have an influence on the resolution. The type of specimen, if there's coherence of the illumination in the system, or whether there's correction of aberrations in the system, that all will influence the resolution achievable. It's important to remember that the rules of the optical resolution applies to conventional microscopes, like microscopes, but the development of the super resolution microscopy does achieve resolution below the theoretical limit of resolution. Also, an important fact to remember is that magnification is not a part of these equations. So it's not a factor when we talk about optical resolution. It's basically only the numerical aperture and the wavelength of the illuminating light that determine the specimen resolution. Just to look at a few examples, this is the head of a zebrafish embryo, imaged at 10 times objective with a numerical aperture of 0.4. If we magnify it a little bit more, this time with a 20 times objective, but a higher numerical aperture, and eventually 63 times objective, 1.4 numerical aperture, you can see a lot more detail in these areas. Let's move on to spatial resolution and digital images. Now, Napoleon Bonaparte said, a picture is worth 1,000 words. We will talk about this now. In the older days, we looked at analog images. These are grayscale images, but today we are looking mostly at digital imaging. Digital imaging means that we are dividing the space into small blocks called pixels, and each of these blocks has a number that relates to the intensity of that pixel. So if you have bright pixels, the numbers are quite high, where if you have a dark pixel like this one, the numbers are quite low. This means that today, thousands of intensity values in space is basically making up a picture. Pixel size is something that affects the spatial resolution. Let's have a look at this example from the mouse intestine. If we zoom in this area, you will see that this image becomes quite pixelated. If we look in the metadata of this image, you will see that the pixels are actually quite large, relatively large. This is 420 nanometers by 420 nanometers. If you manage to image the same area with much smaller pixels, say, for example, 140 nanometers by 140 nanometers, you get rid of some of that pixelation and the resolution is improved. This is spatial resolution being improved. What happens is, if your point spread function is basically circular and you choose a pixel that is bigger or larger than the point spread function, that point spread function is not represented anymore as a circle, but rather the whole pixel block. So the whole pixel block has one value. Let's have a look in imaging the effect. This is an image of a young starfish, which is represented by 175 by 175 pixels. You can see that it's quite detailed, and the pixel size is good, has been selected good enough to see this detail. If you look at the same sample, but this time you divide the image into 10 by 10 pixels, which means the pixels are a lot larger, you basically get rid of that detail that was possible to be visualized. If you want to have a look at more how this pixel size affect spatial resolution, there's an interactive tool that you can use to visualize the changes in resolution. Now, how do we know what the pixel size should be? As I've explained, if your point spread function is circular and you choose a pixel that is too big, representing that one point spread function, you change it into a pixel block. The idea would be to that the spatial resolution must be higher than the optical resolution. In other words, for the point spread function should be represented by more pixels than the actual size of the point spread function. Nyquist's theorem says that the pixel size should be set to about 1/2 to 1/3 of the minimum spacing that one can optically resolve, which means if you have a point spread function or expect resolution of about 300 nanometers, the pixel size should be selected as 100 to 150 nanometers. Then you can really represent the point spread function well enough. Some practical considerations. It's important to remember that making the pixel smaller doesn't necessarily achieve better resolution. Sometimes you actually collect less light in each pixel, which means it gives you a worse image, and it's an example of oversampling. In the examples here on the right-hand side, mouse intestine, you can see these images at the top are represented by 512 by 512 larger pixels, and below at double the resolution, 1024 by 1024, which is smaller pixels. But in effect, you don't really see a big difference. So the question is, do I really need to go at a higher spatial resolution? Oftentimes, it's not necessary, and you need to make that decision for yourself. Another consideration, or a way to make that choice, you might not need the highest resolution, especially if you're not interested in subcellular structures. If you're looking at tissue like the mouse intestine here, often you do not need the highest resolution. Also, there's sometimes a practical reason for undersampling. In other words, choosing pixels that are actually larger than the Nyquist theorem. And that would be, for example, if you had to increase the speed of imaging, or when you want to prevent photobleaching, you do not want to image as much on that image and bombard the sample with illumination. Also, some things to consider is that violet light, which has a shorter wavelength than other colors, doesn't mean that it's always the best choice, even if you want to achieve high resolution. The cameras and detectors are often less sensitive in the violet range, and light with shorter wavelengths often cause more damage to the sample, so it might not be the best one to choose. Also, fluorophore properties are also very important when selecting dyes and labels for the features you want to resolve. So it depends whether there are labels available for what you want to image. If you have the choice, the green fluorophores, which are in the range of 500 to 550 nanometers, usually have a better resolution than red and far red. And therefore, if you can label the smallest features with the green fluorophores, then it's a good choice for those aspects of the image. Thank you very much for listening. These are the people I would like to thank for contributing to this presentation. Thank you very much.